Hello everybody, my name is Miguel Chirivi. I am part of Dr. Contreras Lab at Michigan State University, and today I will be presenting lipopolysaccharide induces lipolysis and insulin resistance in bovine adipose tissue. Starting with some background, the postpartum of dairy cows is characterized to be in a status of negative energy balance that due to the onset of lactation that is just recovered after several weeks. Cows adapt to this negative energy balance by mobilizing fat from adipose tissue, increasing the process of lipolysis. This fatty acid trafficking is also supported by a temporary status of insulin resistance. However, some cows that are not able to overcome to this negative energy balance are under excessive and intense lipolysis across the postpartum period. This lipolysis dysregulation usually is supported by a persistent insulin-resistant status, and those cows are usually found with high levels of NIFAS or BHB. The problem is that those cows are more susceptible to inflammatory and metabolic diseases, and among those inflammatory diseases, it's important to talk about those derived from bacterial endotoxin, especially because postpartum cows are likely susceptible to the presence of LPS or endotoxins in general. For example, heat stress, subacute ruminal acidosis, and periparturine diseases may induce an increased permeability of the intestine that allows the translocation of endotoxin into the circulation, and this leads to different complications. This is known as the leaky gut syndrome, and proof of this is this report that evaluate the concentrations of LPS comparing healthy cows in blue or cows with acidosis. And you can see there is a higher content of LPS in cows with acidosis. Similarly, here is another report that evaluate the concentrations of LPS from 3 weeks before calving until 6 weeks after calving, and they evaluate healthy cows down versus cows with clinical metritis, and you can observe that most of the times there was a higher content of LPS in those sick cows. Similarly, many reports have evidenced the same results in other diseases like mastitis, and as endotoxin is likely present during the postpartum period, we wonder what is the effect of endotoxin on adipose tissue, and especially in this moment of fatty acid trafficking. We wonder this because at least in humans and also in rodent adipocytes, there is evidence that LPS induces lipolysis but also insulin resistance. I have this diagram to present how the lipolysis occurs to make easier the development of my presentation. And normally, lipolysis starts with the stimuli of the beta-adrenergic receptor, which activate the protein kinase A. This one is going to activate downstream some lipases. In these lipases like ATGL or HSL are involved in the hydrolysis of the triglycerides, which leads to the release of glycerol or free fat and free fatty acids. As I mentioned, researchers report that LPS mediate lipolysis by the activation of the toll-like receptor and the activation also of this classical pathway. But also in rodent adipocytes, lipolysis by LPS is mediated by these purple arrows by dependent of ARK1 pathway. And finally, there is an additional pathway reported that mediates lipolysis by LPS, which is dependent of the inflammatory pathway NF-kappa beta and TNF-alpha, which also results in lipolysis. However, we didn't know if this occurs in dairy cows, and that is why our hypothesis was that LPS induced lipolysis and insulin resistance in bovine adipose tissue. We had a couple of goals. The first one was to determine whether or not a peracute and acute exposure to LPS induces lipolysis, and we also wondered if this exposure may induce insulin resistance in bovine adipose tissue. For this experiment, we used 24 multiparent non-lactating and non-pregnant dairy cows. We collect subcutaneous adipose tissue, and the explants were incubated at peracute 3 hours or acute times 7 hours at 37 Celsius in KRBB. The explants were exposed to different treatments. The first one is without the addition of any reagent in order to have the baseline conditions of lipolysis. Then our positive control of lipolysis with isoproterenol. Iso is a beta-adrenergic agonist. Then we have our treatment of interest with LPS derived from E. coli, and then the combination of both. Going to our second goal, we evaluate the anti-lipolytic effect of insulin alone or in combination of ISO, LPS, or LPS plus ISO. The rate of lipolysis was determined by glycerol quantification in the media, and we use also the adipose tissue for gene and protein expression analysis. Our data was analyzed using mixed model from SAS, and the incubation time 3 hours and 7 hours was included as a repeated measure within the random factor count. Finally, we took a post-hoc adjustment for doing pairwise comparisons, and going to the results with the first question, if does LPS induces lipolysis in bovine adipose tissue, the answer is yes. This is graphs representing the content of glycerol, which reflect lipolysis, in nanomole per milligram. At 3 hours, the content is in the white bar, and then the top of the bar is representing the total content at the 7 hours. As you can observe, even at the 3 or the 7 hours, there is activation of lipolysis by ISO, LPS, or LPS plus ISO comparing with basal conditions. This graph is also represented here, and you can observe that most of the times in the treatments present a constant lipolytic rate across the time. Then we wonder how this process of lipolysis is activated. And we evaluate the phosphorylation of the HSL, hormone sensitive lipase, because the phosphorylation of this protein, this lipase, is going to reflect lipolytic activity. Then, as we expect, when ISO was present, there is an activation of this lipase at the 3 or at the 7 hours time points. However, when LPS was included, there is just activation of this lipase at the 7 hours. And then our question was, how then is lipolysis induced here at the 3 hours if HSL is not involved? And that is why we evaluate another second lipase, which is not usually reported in cows, and it's ATGL, and less in bovine adipose tissue. And we found that at the 3 hours time point, ATGL is mediating lipolysis by LPS. Then we wonder if does LPS induce insulin resistance in subcutaneous adipose tissue? And the answer is yes. This graph presents the percent of the reduction of the lipolysis when insulin is included. And as we expect, when ISO is present, insulin reduces the lipolysis by 60% and by 30% at the 3 or at the 7 hours time points. Then when LPS is present, insulin just reduces the lipolysis at the 3 hours time point, and this anti-lipolytic effect is disappeared at the 7 hours. So then we start talking about here about a process of insulin resistance. Finally, when LPS and ISO are present together, there is not activity of the insulin in the reduction of the lipolysis. Also, as insulin is able to reduce the lipolysis and is able to reduce the content of HSL, when insulin was present, HSL was reduced just at the 3 hours time point when LPS was present. But then at the 7 hours, you can observe that there is activation of the HSL. Then insulin was not able to reduce this lipolysis activation at the 7 hours. Then we wonder how this process of insulin resistance may occur. And we target one important protein that is involved in insulin signaling, which is AKT. AKT is important because it mediates the principal biologic functions of insulin in the adipocyte, like the reduction of the lipolysis or the increment of the lipogenesis. And the phosphorylation of this AKT is going to reflect insulin activity. Then, as we expect, there is a higher phosphorylation of AKT when insulin was in the explants. And similarly here, there is still an activation when insulin and ISO were present, suggesting that there is still a process of the reduction of lipolysis when ISO were present. However, when LPS was present in any of the combination of treatments, there is not activation of the protein AKT or the insulin signaling, suggesting that or confirming that there is a process of insulin resistance. The conclusions, we have that LPS-induced lipolysis by adipocyte triglyceride lipase during the peracute time, or 3 hours. Then during the acute time, HSL, hormone sensitive lipase, is the responsible mediating lipolysis by LPS. And we also proved that during acute exposure to LPS, there is insulin resistance in bovine adipose tissue. In general, those results suggest that dairy cows with endotoxemia may be more susceptible to dysregulation of fatty acid trafficking, basically by increasing lipolysis and insulin resistance. For future directions, we would like to define the mechanisms by which endotoxin induces insulin resistance in bovine adipose tissue, and we already identify some important cytokines that may be involved. So I would like to thank to Dr. Contreras' lab team, also to the funding support from the College of Veterinary Medicine at Michigan State University, and finally to the USDA for funding this project. Finally, I would like to say thank you very much to the Tri-State Dairy Nutrition Conference to give us this opportunity to share this research. So thank you so much again, and I will be happy to answer any questions.